So I'll try to weave in how I got into this along the way, but basically the short answer is that we know that a lot of our variables are confounded that we care about, and the big confounder is some sort of innate endowment. I was inspired way back in '96, I think. Susan Mayer, sociologist at the Harris School in Chicago, came out with a book called What Money Can't Buy, Family Income and Children's Life Chances, in which he basically said that all the poverty research was wrong because the effective income on children was vastly overstated. She did a number of quasi-econometric little tricks, but then, hanging over that, we can do econometric, use the natural experiment approach to try to estimate the effective income. However, we also know that the big confounder is probably something like ability, and that's probably, we know, got a genetic component, and so that's how I kind of got interested in this. That's the short answer. But let me talk a little bit about sociology in general and what molecular genetics might inform within sociology. So I'd say probably the central concern, I hear there's 9 sociologists in the crowd, so you can feel free to correct me if I'm wrong and or add. I mean, there's gonna be a lot. I'm not gonna talk about like religion, sociology of religion and culture, but here's, at least in my neck of the woods, some of the big issues in sociology. The biggest is inequality. There's lots of research on cross-sectional inequality, group level inequality, but a really big literature on intergenerational mobility going back at more than 50 years. Father-son at one time, now parent-child, and as I was just saying, we think that genetics probably plays a big role in that, and we at least want to factor it out. Social cohesion versus conflict versus change. I was once told when I was a grad student that all of sociology could be boiled down to inequality and equality on the one hand, one axis, and social cohesion and conflict on the other. I don't know if that's true, but networks and organizations, social movements, and revolutions are objects of study in sociology. In this area, I'm not really going to talk about this today, but I think you could imagine how genetics might infuse this area of research as well. Of course, race and ethnic relations, and included in that is issues of migration and assimilation or lack of assimilation, and pluralism. And I will mention some research of how genetic data can illuminate this area. And lastly, demography and the family. I will talk about assortative mating and fertility and effects of genetics on that. So why should sociologists care about genetics? We were founded as a discipline pretty much against biological explanations in the 19th century. So it's a little bit of a dangerous step to take to reintegrate genetics or biology into sociology. But obviously some of us think that in order to have a totally complete, or at least the most complete possible view of human behavior, model of human behavior, you have to kind of get beyond the nature-nurture debate and integrate them. So as Dan already talked about, simply as a control variable, we can reduce emitted variable bias. We can look through the historical record that's encoded in your genome. I'll show an example of that to talk about sexual interaction or reproductive histories of different populations. So for example, I won't talk about this paper, so I'll just mention it quickly. I have a paper that looks at, as I'm sure most of you know, that for those who are of Eurasian descent, 1 to 3% of our DNA is of Neanderthal origin through reproductive interaction with Neanderthals. And what are the effects of that today? We actually find in the HRS that there's positive effects. We can find some positive effects, but more importantly, you can actually just read through different parts, in particular the X and the Y chromosome and mitochondrial DNA. You can see what kind of sexual interaction was taking place in ancient history, whether it was which group was more the males introgressing their DNA into the female gene line. So Dan's already talked also about this, that yes, any individual marker or even gene will have a pretty small effect, we think, for most behavioral outcomes in the general population. However, when we sum those effects, we can actually explain a decent portion of the variance, and we get pretty big effect sizes if you think of it in standard deviation terms. And then I would say that the big payoff, that at least for sociology, is the notion that we can get from the adage that if there is a gene for aggression and you're born into the ghetto, you end up in jail, but if you have that same gene and you're born in Greenwich, Connecticut, you end up as CEO. The idea that, yes, we come with endowments, but the context matters, and the intuition is that the genome will provide the prism of sorts to refract the white light of average treatment effects into this rainbow that we can actually detect, why we see that if you do peri-preschool or some reading intervention and you get a 10% treatment effect, actually that's probably a few kids with a doubling of their reading scores and most kids with no effect. And that, again, the intuition, although we're still in the very infancy of this science, is that the genome will be the prism that tells us which kids get that huge boost from the intervention and which kids don't get any returns. And and a more formalized theory about that is what's called the stress diathesis hypothesis. You might have heard it termed orchids versus dandelions, that some kids are more sensitive to their environment, they're the orchids, and some kids are less sensitive to the environment, they're the dandelions. And I think Coulter's talking tomorrow, so he probably will cover that, I'm guessing. Yeah, okay. And that if we know biological or neurodevelopmental pathways or even behavioral pathways that are revealed by genetic analysis, we can actually better design policy interventions. The problem with G by E, even though that's the holy grail, I think at least, is that it then becomes an order of magnitude harder in terms of estimation, because it's hard enough for us social scientists to get an exogenous E, some natural experiment, I'll talk about a couple, or an actual intervention, but then also to have randomized G, to say that the genome, whatever measure you have, whether it's a polygenic score or a single gene or some other haplotype, is not just picking up, is environmental confounds. So you've got this double whammy you have to solve. Of course, I think we're making a lot of progress in that regard. So I'm going to run through this really quick because you're going to talk about this ad nauseum for the remaining 2 weeks. But most of the chips that capture genetic data for these large data sets like HRS, AdHealth, UK Biobank, and so on, are what's called Snip Chips, single nucleotide polymorphisms, where there's a single base change, and usually there's only two alleles, in this case A and T at this particular site, and it captures about 1 million of those. You can then impute more if you want, but those are the ones that I'm going to be talking about for most of the work going forward here. So a lot of interest in sociology and developmental science started, well, let me back up. My interest, as I mentioned, started with the Susan Mayer book, and finally, 20 years later, we had this measure, thanks to Dan and David and the Social Science Genetic Association Consortium, SSGAC, that we can actually, instead of having a latent measure of something like the genetics of educational attainment, we can actually have a measured score that we can assign to people, and we can then use that as a moderator and a mediating variable. And so we took the first version of the score and we looked at two data sets, HRS and Framingham. We also did a Minnesota twin study, Minnesota twin family study, and we basically asked a very simple question of, if we put the score in, how much does the effect of parental education on offspring education change? And this is what Dan was referring to earlier as the G as a control variable to get better estimations with confounding. And as you can see from this was the earlier score that explained about 2.5% of the variation. And if you put parental education in, that explained about 13% of the variation. And when you put them both in, it really was additive. One was, at least in this measure, one was not confounding for the other. As you can see, probably better in this chart. Mother's education went down slightly. Father's education went down not at all when you controlled for the genetic endowment. And when we did some calculations, taking into account the enormous measurement error, since we had a very poor measure of the genetic endowment, explaining 3% of the variation as opposed to, say, 20%, we calculated that maximally 1/3 of the effect of parental education was actually not environmental, but was actually genetic in these models. And you could do the same thing with income. You could do it with all sorts of intergenerational models. I'm going to race through these examples. Dan and David suggested maybe I should go through fewer in more depth, but I thought, since it's just the beginning, I kind of want to show you like what's possible out there. Principal components are of the genetic data are often used as a control variable, but they also indicate ancestry. So this is add health. Maybe, I don't know if in the worksheet in problem set 5 you're actually going to do this, but you could do this, which is take self-reported race and plot it against the first two principal components in the genetic data, and you'll see that, for example, African Americans, which are the black dots, vary enormously on the first principal component, which is generally considered an indicator of African ancestry. You can see that the race is socially constructed in the sense that there are some folks who are self-report black, who are to the left of some self-reported whites on the first PC. If you see those gray dots are the whites there on the top, at the top. You can see that Latinos are a mixture of European, Asian ancestry is considered PC2, and African ancestry. There's an interesting mix there. They're scattered all over. Very few Native Americans in Add Health. So again, just this is what is continental. How does continental ancestry map onto self-reported race? It doesn't. It's number one, it doesn't predict perfectly. Number two, you can see there's a lot of heterogeneity in ancestry, even within these categorical distinctions that we use in the United States. Go in a little deeper. This is from Guangguo's work that was published in Demography. You can look at different Latino groups, for example. You can see that Mexicans in the lower left have more Asian ancestry, and that if you look at the top, you can see that the average European ancestry for African American is about 8 to 10% in Add Health. Maybe some of you saw the recent article in the New York Times that cited a PLOS One article that looked at particularly the Great Migration and and African ancestry, and how African ancestry decreased as you move north among the U.S. black population. So there's lots of history you can understand through reading the genome. Here is another example from Ecuador, where there are the native indigenous people, there was African slaves brought over, and then there were, of course, the conquistadors, and very little was known about the how the mestizo population was formed. And here, actually, they found that there was much more equality when they looked at the X chromosome and the Y chromosome admixture in the three groups. So in other words, the assumption had been that basically the conquistadors were male and had, through rape and terror, passed their genes on to the local population. But actually it was a much more complicated story. So this is something that you could try to do through traditional history, but certainly genetics adds an interesting set of data that you can examine. Okay, so here is what I think is the as I mentioned before, the most important payoff, and that is gene-environment interaction. If you have this is what I was saying before, if you have the gene for aggression, you live in the South Bronx in the 1970s, you end up like Medea in jail, and if that's Mitt Romney's house, then you end up in the boardroom and then jail, I guess. So, and a lot of people who are interested in G by E got into one of the sort of seminal papers was published in Science, actually two papers by the same group that followed the Dunedin cohort from the South Island of New Zealand for 30 plus years. And they started with candidate genotyping, where they looked at a very specific locus, that is the promoter area, one of the regulatory regions for the serotonin transporter. And that was based on animal models and what we knew about the biochemistry of depression. And they found what they called a gene by environment interaction. That is, if you had the long promoter, there's a long allele and a short allele. So if you were, as you can see, the bottom line are those folks that are homogeneous, homozygous for the long, and the top group is the homozygous for the short. The short is supposed to generate less expression of the transporter, which is a little bit counterintuitive if you understand why you might be taking Prozac. But and then the heterozygotes were in the middle. And what they found is that if you had this idyllic childhood, or you weren't maltreated at least, then there was no effect of your genotype. But if you had you were maltreated, abused, or neglected, then those who had the long alleles still weren't affected, but those who had the quote-unquote risky alleles, or what they call the orchid alleles, going back to that language, actually had very high rates of depression in young adulthood. Now, as you can imagine, the problem with this study, even though it stimulated an enormous amount of interest and research and replication efforts, and there's huge battles going on between the Caspi et al. Group and Risch, have done competing meta-analysis of since then. I haven't even kept up. I'm sure it's still going on, the fight. The last I checked was, I think, 2009, and there were major fights about whether or not this replicates or not. I would put my money on no, and even if it does replicate, the problem is the fundamental endogeneity of the environment. So I'm guessing it's not that New Zealand has a policy of randomly going around mistreating certain kids, but actually that it's their parents, and their parents are not only passing on that culture and that experience, but also 19,999 other genes that they have not measured. And so we have no idea whether this is an epistasis, whether or not it's a gene interaction or a gene-environment interaction, or what's going on. So that was the problem with a lot of the early research. Here's one from Sociology, again from Guangguo, who did the Ad Health race study, and where they looked at the effect of the dopamine receptor 2 on whether, and the environment that they interacted it with was whether or not the parents had regular meals with adolescents. This is actually, you could reproduce this. This is Ad Health data. And they found that when there was no family meals, regular family meals, there was increased risk of delinquency, and there was no effect of the gene in the maroon bars or red bars when there was a regular kind of socially organized household where there was family dinners. Now, of course, again, I probably can't think of a more endogenous environment than whether or not you have family dinners at home. Since I don't think I probably have one family dinner with my kids every month, I really hope this is a spurious effect. All this early work was really whet the appetite, but didn't really meet what we would, we even knew back then was the standards of modeling environmental effects. So really, we got to focus on the exogeneity of E. I'm going to show you a couple examples from my own work. What we did, this is still in the candidate gene era, was that we used this is also Ad Health. We looked at the markers, and this in particular, this is DRD4, the dopamine receptor 4, and we use the logic that among dizygotic twins, basically full siblings, it is a random assignment at conception whether you got the quote-unquote risky allele or the quote-unquote dandelion allele. Right, so there we have an exogenous G, but in order to do so we could find a main effect using sibling fixed effects of the gene on the outcome. Great, I think that's a pretty good design. It's with the policy experiment of randomizing at conception the genotype. However, in order to compare the environment, we have to compare across families, across sibling sets, essentially stratify them by some sort of environmental difference. What I look for here is birth weight variation. This is work by myself from 2000, and then in economics since then has shown that birth weight is a pretty good proxy for prenatal endowment, for prenatal environment, and it has effects on academic outcomes and socioeconomic attainment as well as health. So we use birth weight differences between twin sets. So when we have monozygotic twins, we know that the birth weight difference is random, is totally environmental. It is just who got the front seat and hogged the placenta and who got screwed in utero. But the problem is then their G is exactly the same. So we have an exogenous E, but we have G has to vary across twin sets. So and then go back to the dizygotic twins, we have G is randomly assigned at conception, but E not only picks up the uterine environment differences that they experience, but also potentially genetic effects. So that's confounded because we're not measuring all the genetic differences on their birth weight. So strategy is basically do the best we can and do it both ways. Use the dizygotic twins where G is randomized for sure, and use the monozygotic twins where E is randomized, and can see if the estimates match. Then we feel more confident. So what we found was that that with the quote-unquote risky gene, that an additional pound of birth weight led to basically 3/4 of a grade point better GPA. But for those who had the normal gene, birth weight did not affect it. I don't believe these effects. Speaking of you can throw the underpowered tomatoes at me now, Dan. But this is really proof of concept of what kind of data you need to make the claim to make a claim of exogeneity. So we get the exact same effect whether we use the DZ strategy or the MZ strategy. So we think whatever, if it's a false positive or whatever, at least it's the design. Yes, we would love to have 10 times as many folks. We would love to have a polygenic score, which we will have, and so forth. But at least it's proof of concept of you could use birth weight differences and sibling differences in genotype in order to estimate something like this. So to summarize the G by E stuff, Most G by E studies, as I mentioned, are problematic, and I just don't even bother reading them to the end because they use endogenous measures of environment. So we don't know if there's underlying genes that are selecting people into their environments, what are called RGE, and therefore proxying actual G by G interactions, epistasis. So examples of exogenous forms of E that's been used by myself and others, birth weight differences I just showed you, policy changes, the compulsory schooling law in the UK that Dan mentioned before. I've used the draft lottery, I'll show that in a second. Societal shocks, other people have used the Great Recession, earthquakes, literally, and even 9/11, because Ad Health was in the field in 2001, and half the interviews were done before 9/11, half of them were done after 9/11. Other people have used peer, there's a study by Guangguo that uses random assignment of roommates in college, freshman year college, to look at genetic environmental moderation on binge drinking, and so forth. We want to move beyond the candidate genes where we know that explains very little of the variance, and to the polygenic score approach. I'm going to skip through this because you saw this equation many times already, and you'll go through it much more in depth when you calculate the polygenic score. But this is the original paper for education, where it explained about 3% of the variation, almost 3% of the variation when we took a single score. The new one is about 7% in Minnesota, HRS, Ad Health. And so the idea is to redo this approach and use the polygenic score as opposed to a single gene. So these are the old slides. Oh well. So you could see that this is the old score is correlated just below 0.2 with years of schooling as compared to, say, BMI or height, which do better, but it does better than a lot of the medical scores. And now we're at a higher correlation. So smoking, diabetes, depression, heart disease, okay, small correlations. Alzheimer's and psychiatric disorders, pretty bad. So, okay, so using the polygenic score, you can also integrate that with a traditional natural experiment approach. So this one is the Vietnam draft lottery. Happened to be that HRS, Health and Retirement Study, had the right birth cohorts that were exposed to the Vietnam draft. This is the guy I consider well, at least up till that point, the dumbest politician ever, because he thought that picking the ping pong balls with the birth dates on it, you couldn't vote at 18 at that point, but the parents of these people could vote as he was sending them off to be drafted and go to fight in Vietnam. I don't know why he thought that was good TV exposure. I think that's Robert Moses to his right. He looks like it. But anyway, this was the 1969 draft. So you can't get a better sort of randomization, except for one of the drafts where they didn't stir the balls enough. But if you just do month fixed effects, then you're all right. So we took the draft number, the draft lottery status, and interacted it. We use it as an instrumental variable to look at veteran status, and then saw if the effect varied on smoking behavior. We know that the Army was a very tobacco-philic, whatever the term is, environment. They literally gave cigarettes to the GIs to keep them awake when they needed to. Is that a question?
Yeah. So does your draft number depend upon your month of birth as well as your year?
In one draft, if I think it was 69, they didn't stir, they dumped the balls in, they had them 12 crates, and they dumped them in, and they didn't stir them enough, so there was a month effect. But if you put month dummies in, you get back to random.
Okay, great.
So, and we show that it's not sensitive to that.
I mean, excluding that year. There's also a nice, the 70 draft, I think, or 71, the last draft, they did the draft, and they didn't actually call anyone up. So it's a nice counterfactual to make sure that there's not another pathway violating the exclusion restriction, like employers are discriminating against people with bad draft numbers or something like that, which was a critique of Heckman. We can talk about that in the breaks if you want, but that it's been, since the original Angrist paper in 1990, it's been through the ringer a lot. I think it's generally accepted as an instrument at this point. So we looked at smoking behavior, and there's a polygenic score, as I showed, that predicts smoking behavior. It's not great, but it has some R squared. But what we looked at is whether or not the effect of being drafted or being a veteran had on your lifetime smoking behavior varied by genotype, and it appeared to, so that if you were at 4, I mean this is very extreme, but 4 standard deviations to the right in terms of your genetic propensity to smoke, you were basically the difference between a veteran and non-veteran was 2 packs a day, and at the bottom end, as you can see, there's no difference. So this is, again, I'm going to police myself about power. Ideally with a draft lottery study, you want the census or some data set with millions and millions of people. We have a few thousand. We have, in the HRS, you have about 10,000 overall genotype folks. When you get down to the right age group, we're down to, I think, slightly over 1,000, if I remember correctly, and probably underpowered. So this is almost like an advertisement for, these are the kind of exciting things we could do when we have really large data sets to cover a lot of, but again, proof of principle is what we should be doing. We should be using natural experiments or interventions for E, and in this case, we should be using polygenic scores. This controls for those principal components that I showed you before to take out ancestry effects that could be confounded with the environment. So another approach you could do is just say, look, I'm going to look at how the genetics penetrance for a particular phenotype changes over time. I know that the environment's changing over time, probably not the genetic makeup of the population all that much over time. So if this is probably a G by E interaction, if I look across the 20th century and see that the effect of a particular genotype is changing. So here we see that in line with other studies that have used twin models and you've used other approaches, that the polygenic score for smoking increases in predictive ability or increases in effect across the birth cohorts of the health retirement study. There are a lot of issues about mortality selection and so forth that I'm happy to talk about, but don't really affect the results. But this makes sense. Other economists have found that the price elasticity of smoking has decreased markedly over recent cohorts. Cigarette taxes no longer have as much of an effect. So the theory is that the residual group of smokers are smokers that are really genetically hardwired to smoke and very unelastic to policy. So we find this using the polygenic score as well. There's lots of other examples. So for example, what would you expect in terms of height and body mass index? We went from the early 20th century cohorts where there was still caloric restrictions for a lot of the population to the land of post-war McDonald's. And what we find is these two lines, these two with the error bars, are the group, the predicted value for someone who is 1 standard deviation above the mean in their polygenic score for height and for BMI, and the bottom line is someone who's 1 standard deviation below the mean on their polygenic score, and you can see that the gap gets bigger between those two lines in more recent, there's probably a nicer way to represent this, but the interactions are significant. So as the environmental restriction, this is the theory at least, as the environmental restriction on our calories is uncapped, then we get a full flowering of genetic difference. Well, what about education? Education, actually, you would think, okay, educational opportunity, maybe the same logic holds for educational opportunities. Before you had people who were held down on the farm and they had this incredible ability, but there was no schooling to go to. And then now we have access, almost universal access to schooling. So that we should see the same effect where we see increasing genetic penetrance over the course of the 20th century. And that's what some twin studies found earlier. We find the opposite. It's very marginally significant, but at least we find we can reject the one-tailed test pretty well, that there's not increasing genetic effects over the course of the 20th century. And if you guys have read the Nature paper, was that assigned, I assume, that finds the exact same pattern in Sweden as well. This is the US, so that there's decreasing genetic penetrance. So that was contrary, I think, most of our expectations, which is interesting. We should be confounded by, have our expectations confounded sometimes. So last thing I want to talk about is the family and assortative mating. So you see my subtitle here is it safe to assume that G is exogenous for cohort studies? So I've been showing you these trends in genetic effects, and the assumption is that, oh, whatever is changing that's the environment changing. Well, wait, what if actually the genetic makeup of the population is changing as well over the time? Well, in terms of like new mutations, probably not a huge amount over 4 generations, but what if there's differential fertility and assortative mating? That can change the variance and the mean levels of the genotypes. So I have like 10 more minutes. So you'll learn how to calculate these, the relatedness coefficients, but basically it's a correlation at each locus of if you have a million marker chip, and you can sum those by chromosome or by entire genome and estimate how correlated everybody in this room is with each other on the base of this population. So you're going to do Gremel, I assume. So that you'll do that. So what we did is we looked at basically in a society like the United Arab Emirates or Pakistan, over half of marriages are between first cousins. What is it like here? We think that we are in a modern, highly mobile, ethnically based society, modern society. Well, we found that the genetic assortative mating still occurred, and in fact occurred to the level of, if you don't account for ethnicity, the average pair of spouses is genetically related as first cousins once removed. If you account for principal components, that is for common ancestry, that maybe Italians marry Italians and so forth, it goes down to second cousins. So before we cast aspersions at my colleagues in NYU Abu Dhabi, where everyone's marrying their cousins, we're actually kind of doing it too. Another question was, okay, so yes, we're slightly more genetically related to people we marry, and also to other people have shown to our friends as well. There's a genetic correlation. But so what? That's across the entire genome. There's like millions of markers. Some are positive, some are negative. Who knows what's going on? Well, we looked very specifically, now that we have these scores, at spousal similarity at the genetic level. So on the left, the lighter bars are the phenotypic correlation between spouses, and the darker bars are the genotypic correlation in terms of the polygenic score, which of course is measured with error, but we found that actually these were at the old scores, and when we redo it with a better score, it doesn't change. So it's something that even though you're measuring both things with error on each side of the equation, the estimates are tighter, but they don't. The point estimates don't change. Didn't change. So you could see that for education, there's an incredible level of assortative mating, as we know. In the United States, it's over 0.5 correlation. But actually, at the genetic level, it's only about 0.15. Height is really interesting because for height, yes, we're sorting slightly at 0.18 on the actual phenotype. We're assorting to a greater degree on the polygenic score that predicts height, which suggests something worthy of investigation, that perhaps height, the polygenic score is pleiotropic and is reflecting general health or robustness, or something else that spouses are sorting on. Because obviously we're not sorting on, not yet, 23andMe and Tinder are not linked yet, but they're obviously sorting on some phenotype that's predicted by the height score, but is not height itself. Yeah.
In the bell curve revisited paper, I think you estimated how the genotypic correlations between spouses are changing over time by regressing the one spouse's polygenic
Right.
score on the polygenic score. Did you do the same thing here?
Yes.
Right, thank you.
So you see that for BMI and depression, we don't see any correlation between spouses. I think we've done it both ways. We just do correlation and we do a regression approach.
Yeah.
So next question is, yes, Dan.
Two questions about, so what was the power on those analyses?
What's the sample size or the actual power? I can tell you the sample size.
Do you think they were reasonably powered?
Yes, for this kind.
All four phenotypes?
Yeah. We have about 8,600 individuals, about 4,300 pairs for these individuals. For G by E over time, you could argue that we're underpowered maybe, but for simple correlation between the spouses on the scores, I think we're adequately powered.
And then on the previous slide, so it seems like a shocking claim that in the U.S. people are marrying people as related as their second cousins.
You mean, I think the first cousin once removed is not that shocking when you think about intra-ethnic marriage. That yes, the interracial marriage, for example, is very rare between blacks and whites, for example. So we're segregating that way. I think it more shocking is that once you control for that, you still get it's the equivalent of second cousins.
And what's your interpretation? I mean, do you think it's really the case that people are largely marrying their second cousins?
No, I don't think they're actually marrying their second cousin. But I think that's probably the sum of these things, sorting on phenotypes, is generating an overall signal, plus probably some residual ancestry and geography that matters. We tried actually controlling for, because in the HRS, there's not detailed self-reported ancestry. There's race. We can factor that out. We factor out ancestry through PCs, and we also looked at geography, state. So, but we can't knock it out with that. But there's probably some, local. I mean, we definitely know that there's local genetic effects. There's a different paper that shows that, for example, polygenic scores and even scores on PCs vary by region and state in the United States.
Sorry, so what is the analysis that led you to conclude that people are just naming people's spouses are as close as second cousins?
We looked at, so the shaded area, so if there was random mating, there would be, this is kind of like a Gini coefficient. So there's a deviation from random mating of 0.045, 4.5%. So that is equivalent of first cousins once removed. First cousins are 12.5% related. So, and again, it goes down to 2.5%, 2.2% if you control for PCs 1 through 4, and that's the equivalent of second cousins. terms of
This is like
meiotic.
basically looking at the average correlation across the genome and genotype.
Exactly. Exactly what you would basically use on the right-hand side of a Greml. So, so we look at fertility, number of children ever born. As you can see, there, as we know, there's a negative correlation between education, phenotypically, and number of children ever born. That's concerned some folks who have been called dysgenecists, very controversial scholars such as Richard Lind in the UK. But when we look at the genotypic level, which would actually have the effect on the average cognitive ability or educational attainment of the next generation, we see, yes, there's a slight negative association between the education, PGS, and number of children ever born. But as you can see, it's nowhere near as big as, it's very close to zero. And we didn't find any evidence that it's increasing in trend as the education phenotypic relationship with number of children ever born actually has been getting larger over time, more negative over time. It was actually positive at the beginning of the 20th century, and it flipped sign in the mid-20th century and has gotten more negative. But we do not see that trend with education. You can see that for height, there's actually a positive correlation with number of children ever born. Again, I think this health endowment issue in terms of the genotype, even though there's a negative correlation for tall people at the phenotypic level. So again, the kind of interesting thing you can do with this sort of analysis. So just to wrap it up, that I think that the way forward for the G by E design is to use the polygenic score just to maximize, unless you're interested in a very specific pathway in which you'd use a subset of genes or something like that very specifically. But if you're interested in just sort of the your general genetic endowment and how that might vary by environment, the best approach would be to use a polygenic score. If you can, within family, AdHealth has siblings and has genome-wide data, so does Framingham, so does a few other studies. Health and the Wisconsin Longitudinal Survey is going to come out with genome-wide data soon. There's a bunch of the studies in Europe and in Australia that have sibling design and also have genome-wide data. Find the exogenous E. Something that I don't know if we're going to talk about is to do GWAS, to look for genes that don't just regulate the mean level. So in other words, if you have allele A, you're taller on average than allele T at that locus, but actually genes that regulate, or loci that regulate the variance, where group A and group B are exactly the same mean level height, but group A, which has this genotype, has a wider variance in height than group B, which has a more tightly regulated height genetically. And I think that will be really, that's just starting that kind of work, and I think that's going to be really important because as we want to look for G by E interactions, because you want to look at the folks where there's a lot of variance, because it's not, they're probably more plastic to the environment than ones where they have a genotype that everybody ends up exactly 6 feet tall. And I think cohort designs are promising, but there's a lot of caveats to that, where you have to be concerned about mortality bias, about attrition, and again, whether or not for your particular genotype and phenotype, the underlying distribution of the genotype is not changing, because that's the kind of key assumption that's constant over the 20th century or over some time period if you're going to look at cohort differences. So I'll stop there. Thank all my collaborators.